Good morning. Thank you for the intro and having me here today. Again, my name is Julianna Proietto. I'm a senior animal specialist at Hackensack Meridian Health Center for Discovery and Innovation in Nutley, New Jersey. And I'm excited to share with you today the preparation of agar-bead-embedded Mycobacterium abscessus to inoculate immunocompetent mice intratracheally. Mycobacterium abscessus is an environmental bacteria that can cause pulmonary infections, particularly in immunocompromised individuals and with underlying respiratory diseases such as cystic fibrosis. It is known for its multi-drug resistance, and treatment typically involves a combination of antibiotics, often requiring prolonged therapy to effectively combat the infection. There are limited treatment options for MAB pulmonary disease, and the development of a new drug that is safer, shorter, and more effective is urgently needed. Preclinical evaluation of antibiotics against MAB infections has proven to be challenging in mice because establishing a progressive and sustained infection in mice has been difficult. Antibiotic evaluation in mice is challenging because infection of immunocompetent mouse strains with MAB largely result in transient colonization followed by rapid clearance of the pathogen. In the standard MAB infection, which is on the left, the mice have an innate immune response, making this model unsuitable for a chronic infection. This brings us to the issue of needing a chronic infection model for drug development. Embedding MAB in agar beads enables prolonged infection by protecting the bacteria from innate immune clearance, making this model suitable for a chronic infection. The current study was performed to generate reproducible batches of agar bead embedded bacteria to ensure consistent MAB-loaded agar beads and bacterial burden. This leads us to achieve reliable intratracheal inoculation and lets us establish a chronic infection in an immunocompetent mouse model. Here, we want to provide a detailed protocol to reliably achieve these objectives and ensure lab-to-lab reproducibility. Next, I would like to discuss the preparation of agar bead embedded MAB inoculum. This method was done by our microbiologist Wathahoon. I will discuss the major parts of the preparation. So we prepared a pre-cultured Bacteria abscessus and proceeded to incubate in a roller bottle for 24 hours at 37 degrees Celsius to achieve the mid-log phase. The mid-log phase of OD600 ranges from 0.4 to 0.6. We estimated a mid-log phase of 30 by spinning it down to make a concentrated culture. We then mixed the culture by pipetting it with molten tryptic soy agar. Following that, we emulsified the bacteria in mineral oil and stirred it at controlled temperatures, beginning with stirring for 6 minutes at room temperature, followed by 35 minutes on ice. We then proceeded to our washing steps to remove oil and free bacteria. Once we had a clean sample of only agar beads, we filtered them to achieve the target size of 150 to 250 micrometers. We then tested 6 different batches of beads that were made. These results show a reproducible bead preparation across multiple trial runs. We modified the previous studies to minimize the variability of bead size by adding multiple washing steps, decreasing centrifugation speed and time, and we confirmed sizes across all trial runs were consistent. This brings us to intratracheal inoculation of mice. The first intratracheal infection attempt was deemed unsuccessful due to the variability in CFU counts. CD1 mice were initially sedated with ketamine and xylazine, placed on their backs, and secured with a suture string over a rod. We used a plastic gavage attached to a plastic syringe to deliver the agar beads. Despite confirming proper placement in the trachea using a water bubble test, inconsistent CFU results led us to suspect issues with the plastic materials. To further investigate, we conducted pass-through tests by dispensing the bead suspension through the plastic gavage and homogenizing it for CFU enumeration. Results varied significantly, suggesting that the agar beads were likely adhering to the plastic surface, leading to inconsistent infection. We then repeated the pass-through test using a metal gavage. This produced consistent CFU counts, confirming that the metal of the gavage affected bead delivery. We initially used a 22 gauge metal gavage, which passed the beads easily, but it was difficult to introduce into the trachea of the mouse. Based on recommendations from Colorado State University, we switched to a 24-gauge metal gavage, which provided both ease of insertion and consistent delivery. These optimizations led to the final setup, a 24 metal gauge gavage, 3D printed stoppers and stand, and the glass Hamilton syringe. The stoppers were designed to exactly measure 50 microliters, allowing a total of 100 microliters of agar beads to be administered in 2 doses, giving the animal time to recover between doses. We also adjusted our methodology based on insights from our collaborator at Colorado State University. We discontinued the use of ketamine xylazine sedation due to its respiratory suppressive effects and adopted a ventral recumbent positioning of the mice, which offered improved access to the trachea compared to the initial supine setup. These combined changes significantly improved the consistency and success of the intratracheal infection model. We then tested intratracheal inoculation of 10 mice with 3 different operators. The mice were euthanized at 24 hours post-infection, lungs were collected, and homogenized for CFU counts. From the graph, we can see all samples were relatively consistent with just 2 outliers. With confirmation that our protocol is reproducible, we explored a larger infection. These are the CFU kinetics and lung histology images of C57 black 6 mice that were infected with MAB intratracheally. Lungs were sliced via the Leica 1860 cryostat and stained with zeolnicin. Day 14 and day 21 lung histology show a high density of bacteria in the macrophage-rich center of granulomas. The image at the bottom showed an early necrotizing granuloma and foamy macrophages with intracellular bacteria. These results confirm that infection with MAB-embedded agar beads can be used in chronic in vivo drug efficacy studies. The major limitation of this protocol is the technical complexity of the bead preparation and the intratracheal inoculation to achieve reproducible bead burden and lung implantation. In order to overcome these limitations, we paid attention to detail on orientation of the mouse for infection, sedation procedures, and the reasons why we had inconsistent CFU counts. The advantages include the use of fully immunocompetent and most affordable C57 black 6 mice, which is a more humane method compared to other methods. Other methods involve the use of immunosuppressants such as cyclophosphamide and dexamethasone, and these immunosuppressants may introduce drug-to-drug interactions. Productive and chronic infection can be obtained with a well-characterized type strain. To conclude, we established a robust, reproducible model for MAB chronic infection immunocompetent mice. Now it is possible to perform preclinical evaluation of new antibiotic regimens. Our lab is planning to test antibiotics in the near future, and we hope to extend the infection for a longer period of time. The critical factors you must consider are the temperature, oil to agar ratio, stirring consistency, use of glass syringe, size of metal gavage, and the animal positioning for intratracheal inoculation if you would like to adapt this in your own lab. I'd like to acknowledge my PIs, Thomas Dick and Veronique Dartois. I would also like to acknowledge my fellow lab members, Wasahun, Bedelem, and Camilla. The research animals facility for looking after my mice and notifying me if anything went wrong, and Mercedes team at Colorado State University. Thank you. I'm open to questions.
Hi, I'm wondering, are you able to see if you get uniform distribution of other needs into each of the long lobes? And is that consistent, or have you dissected it to see if it's homogeneous throughout?
We're still working on slicing for histology. We actually are now in a phase where we're sending samples to IDEX so they can stain them for us, but we haven't gotten to that much detail about how encompassing the lungs they are in. No. Thank you.